So thank you all for coming out this afternoon for Advancing Lupus Care Through Research and Advocacy, our second annual research meeting for people impacted by lupus. So today you're going to hear from some great speakers talking about the wonderful LRI funded research that they're doing. And then I change roles and talk about advocacy. There's some brief bios and descriptions of all the research in your packet, so I really don't want to get into going through everybody's credentials. You have it all in front of you. I'd like to leave the time for our speakers, so we're going to move on to Dr. Thurman to talk about his work in nephritis.
Great. Thank you very much. I really appreciate the opportunity to talk to you all today about the research that I'm doing. And I'll start just by telling you a little bit about my background. I am an adult nephrologist, which means I take care of patients with kidney disease, primarily adults. Lupus is one of those diseases that sometimes starts with pediatric care and then transitions to adult care. I see patients with all forms of kidney disease, but lupus is both a research interest and a clinical interest, and I have one clinic a week, a half day a week, where all I see is autoimmune diseases and lupus. So there is an intersection of my research and my lab interest and my clinical interest, and I'll bring that out a little bit with the talk today. So I'm one of those people who does experiments in mice, but also sees patients, and hopefully I learn a little bit from each, and I think that shapes how I view the disease. So first, I just want to define what do I mean when I talk about glomerulonephritis or nephritis. This is a very general term. It really just means inflammation of the kidney. And when people say glomerulonephritis, all that is telling you is that the immune system is hurting the kidney, but it doesn't tell you that it's part of lupus or why it's hurting it. And you know, the changes that we see with lupus can occur with other autoimmune diseases. They can occur with infections. They can occur with reactions to some medications. So it is a pattern of injury that we see. It is a disease that is sometimes cryptic when we first see it, and a lot of the challenge is to figure out what the cause is. We were talking about this earlier today. Unfortunately, glomerulonephritis is one of those diseases that probably the normal story is that patients have it for some time before it's properly diagnosed, because the findings with glomerulonephritis are not that distinctive, and it often takes a little bit of work to figure out what's causing it. In patients with lupus, the disease is very variable. Not all patients are affected. It does not correlate with the blood tests that we get, so we can measure autoantibodies and other markers, but they really don't tell us how active the disease is or what the prognosis is. And this makes it difficult because, of course, a patient's primary question is to know what's going to happen in the future, and it's very, very difficult for us to figure that out with blood tests. In some cases, the disease is very indolent and doesn't need treatment. And in some cases, it's changing day by day, and we have to act very, very quickly. And again, one of the big challenges is to try to look at a patient and figure out which of these groups they fall into. About half the patients with lupus have some kidney involvement. And usually how we do detect the disease is we do a couple of urine or blood tests. We look in the blood, and we're looking for markers of kidney function. So if the kidney function is impaired, that's obviously a concern. And we look in the urine, and we look for leakage of protein, we look for blood and other evidence of inflammation. And none of these things is specific for lupus. And again, that's why sometimes patients have the disease for a while before it's diagnosed. Patients are often misdiagnosed as having urinary tract infections, and really only after things carry on and evolve does someone really think, gee, this might be something more complicated, we'd better look at it more closely. The way we look at it more closely, unfortunately, involves a biopsy, which no one wants, and to some degree that's why the diagnosis can escape us for a while, because we try not to do a biopsy unless we really have compelling evidence that we need to figure out what's going on. When we do a biopsy, we do a couple of tests, and we look for inflammation in the kidney, and we look for antibodies in the kidney, and we look for something called complement proteins, which is a particular interest of mine, so I'll tell you a little bit more about that throughout this talk. Even with the biopsy, it's very difficult to know how active a patient's disease is, and of course, no one wants a biopsy more than they need it, so one of the ongoing challenges, even after we've made the diagnosis, is to say how active is the disease, and is the treatment that we're giving the patient working. So this is how we do a kidney biopsy. So a patient stays awake, they lay on their stomach, and we try to get the back flexed, and we take an ultrasound probe and put it on the back, and we can visualize the kidney, and then we put a needle into the kidney, and we try to keep it on the outside of the kidney so that we can get what are called glomeruli. And that's part of the kidney that's affected in lupus. And then we actually usually put the needle in a couple of times because we have several tests to run, and we send these biopsies for these different tests. We do light microscopy, and just in case you're interested, these round things are glomeruli. We do immunofluorescence microscopy where we can look for IgG and complement proteins, and again, these are glomeruli. And then we do electron microscopy where the pathologist can look at very, very high magnification. So when we do several passes with the needle, it's because the pathologist is going to run a lot of tests, and you don't necessarily need all of these to make a diagnosis of lupus, but it really, when you have all three of these tests, there are very characteristic findings that makes it incontrovertible that this is lupus, and we get a sense of how active the disease is. But I will tell you, this is, and I don't want to discourage anyone from a biopsy, but when we do a kidney biopsy, we are sampling 25 glomeruli out of 1 million that you have. So this is the best test that we have, but as you can imagine, that's a pretty small sample. So it's the best we have, but this itself does not tell us the status of disease throughout both kidneys. What are the drugs that we use? You may have heard of a lot of these, but we always control the blood pressure for anyone with kidney disease. And then the standard drugs that we use to treat lupus include something called mycophenolate mofetil, cyclophosphamide, cyclosporin, and of course prednisone or steroids. And these are great treatments that are very successful in the majority of patients, but of course they all have their side effects. So this is the crux of the dilemma. We have a disease that's difficult to diagnose, and we have medications that have side effects, and this is what we wrestle with month after month. Is the patient responding to these treatments? Is the benefit of these treatments worth the side effects of the drugs? So it's a delicate balance. We know that prolonged and aggressive treatment not only treats the disease acutely, but it reduces the risk of recurrence. And every time a patient has a disease flare, our worry is that even if we treat the flare, a little bit of scarring is done. So there's this desire to treat very aggressively up front to prevent that from happening in the future. On the other hand, the longer we treat with these immunosuppressive medications, the greater the risk of infections, the greater the risk of other complications. Again, this is a challenge on a monthly basis. And I think you all know this, but lupus is a lifelong disease. Even if we treat successfully, I always tell patients, you are never going to be free of this, and we will be asking the same questions over and over again. You're going to come back, we're going to look you over, and we're going to say, do you need treatment again? Are we happy with where things are? And we'll try to weigh this balance of, is it a benefit to treat them further, or is the disease quiet? So how could we improve the treatment of lupus nephritis? I break this down into two very basic categories. We could either get what's called biomarkers, so improved methods of monitoring disease activity, and this would allow us to use the drugs that we have in a better fashion. We could really see, is the disease active or is it responding to therapy? And my lab works on this, and we are trying to develop better ways to check how a patient is doing. Or we could develop new treatments, and the idea here would be that the treatments that we have are good, but they are very nonspecific. Wouldn't it be great if we had drugs that really blocked the mechanisms that specifically cause lupus? And that's the subject of the research project I'll tell you about in the next few slides. So here's where I'm gonna bore you with some esoterica, but this is what we're doing. A couple of years ago, we were looking for novel proteins that interact with the immune system, and we used a method called proteomics. And the good thing about proteomics is that you don't need to know what you're looking for. It can reveal things that you didn't know about. And when we did this experiment, it revealed that a protein called annexin A2 interacts with the immune system. And I'd never heard of annexin A2 before, but it came out and we said, okay, this is interesting. It interacts with the immune system. Let's see what we can learn about it. And over the next few years, we saw that annexin A2 interacts with another protein with a kind of an obscure name, and it's called factor H. But actually in nephrology, factor H is a very well-known and well-studied protein. This is an anti-inflammatory protein that the body makes that protects your own body from your own immune system. It helps keep the immune system focused on bacteria and away from yourself. And the way factor H works is that it binds to your cells and it stops the immune system from attacking them. So what we discovered after we identified annexin A2 is that annexin A2 prevents this from happening. So annexin binds factor H, keeps it off the cell, and lets inflammation carry on. Okay. So we did some experiments. I think this is the only slide of mice that I have, but we took this annexin protein and we injected it at high doses into mice, and we looked at what does it do in the kidneys. And what we found, and this is the experiment where a mouse was injected with annexin, and if you remember what I showed you a human kidney biopsy looks like, this to my eye looks very similar to the way lupus looks in the kidney. There's deposition of immune proteins in the glomeruli. So it looks like this protein that we identified promotes inflammation in the kidney. So what is the evidence that this has anything to do with lupus? Well, you know, around the same time that we were doing that work, a couple of people started looking at these very same proteins in lupus. And as I said, factor H is a very well-studied protein, and this is a study from another group where they looked at factor H in a model of lupus, and the long and the short of it is they showed that factor H is important for protecting the kidney, and that if you don't have factor H, lupus-like disease gets worse. And just trust me, these are glomeruli that are more severely injured than this on the left. So, okay, factor H is important in lupus, and it's also important in other kidney diseases. But there were also two other studies that said annexin A2 is particularly overly expressed in both patients and animals with lupus. So again, these were groups working independently of us. We didn't know about them until they published this data. And what they are showing is that annexin A2, the protein that we sort of randomly identified, is expressed in active lupus nephritis, but is not expressed in treated or quiet lupus nephritis. And in this paper, they also looked at a lot of other kidney diseases, and they concluded that annexin is very, very specific for lupus nephritis. They were not looking at inflammation. They were looking at something else entirely. But because we had found all of these results showing that annexin is pro-inflammatory, that really gave us the idea that this is probably important in lupus nephritis and might be a very unique pathogenic mechanism leading to kidney injury in lupus. So our hypothesis at that point was that in lupus, the kidneys make this protein, annexin A2. Annexin blocks that other protein, factor H, and that's what causes kidney inflammation in lupus. What we hope to develop as part of our project are drugs, or I should say drug or drugs. We're trying several different methods that specifically block this interaction. And our thought is that this will stop one of the key events of lupus kidney disease, but would not have any effect on a healthy patient or a patient who had another type of kidney disease. So that the side effects of the medication would presumably be very low, as opposed to a drug like Cytoxan that's going to wipe out your whole immune system, whether you have lupus or not, whether your disease is active or not. So what are we planning to do? First off, we have all of these other people's work and our own, so we do have to put it together and really prove that this protein annexin is involved in the development of lupus, and that's going on right now. We are in the process of developing these inhibitors, and just to sort of go back to what is so great about these LRI grants, if you apply for an NIH grant without everything you need in hand, they just won't fund it because it's considered too high risk. And so that's why a grant like this is so valuable. We have an idea, we have data, but the truth is, how do we develop these drugs? We need someone to take an interest and support it, and that's what we got from the LRI, and that's what we're trying to do right now. And then finally, we will test these novel drugs in models, animal models of lupus nephritis. So again, here's the model as I see it. We have this protein, factor H. Your body makes it. It's a crucial protein for protecting your body from your own immune system. In lupus, the cells of the kidney make annexin A2 that we think blocks factor H from functioning. That leads to inflammation within the kidney. And we are trying to make novel agents that would block A2 and then let this do its thing. A2 goes away, factor H does what it's supposed to do, and everything is good. if we're able to make such an agent and if it works, this I think would be what I would call a more targeted therapy than we currently have. It will not have any effect on the cells that aren't expressing an XNA2. It would not have any effect in a patient who is healthy. You could put a patient on the drug and not be anxious month after month. Do they have the disease? Do we need to continue this? You could err on the side of over-treating and hopefully do no harm to the patient who is already in remission. So what is the goal? It's, as I just said, it's to develop a drug that will block the mechanisms of lupus, but will have no effect on patients who are in remission or no effect on patients with other types of disease. I do admit there are some conceptual limitations to this. And the first is that we are trying to block lupus injury to the kidney. This strategy will probably not get to the fundamental mechanisms that cause lupus. Patients with lupus are autoimmune. We are protecting the kidney, but we're not reversing that autoimmunity. So it is limited in that sense. And then the other thing is that anything that blocks the immune system, you just have to assume will have some adverse effect on your body's ability to fight infection. This, to me, feels much better than cyclophosphamide or Cellcept, but there is no doubt that anything that you do that blocks a normal immune system function carries the risk of infection, and that's something also that we'll study as we develop these reagents. So I want to acknowledge a couple of the people in the lab and, of course, the Lupus Research Institute for supporting this research.
Or can you get that through a blood test?
Yeah, so it's a great question. So the question was, if we're trying to figure out if an XNA2 is playing a role in an individual patient, is that something that we would figure out from a biopsy, or could we get it from a blood test? And I hadn't even gotten that far ahead. I do think at this point the simplest way to do it would be in a biopsy where, just as that group that published the data showed, we could look at the kidney and stain specifically for this protein and indeed see is it there or not there. So that is an advantage in the sense that you could select patients who are the most likely to benefit from a treatment such as this. But the downside, which I think you're getting at, is that you're still stuck with a biopsy as the way to really make a determination. Does this protein show up in the urine? Is it something that we could detect in another way? I haven't looked at that yet, but I think it's a very good idea, and we probably should start exploring it.
Good afternoon. I've had lupus for 14 years now, and I've been in remission twice, and currently I'm flaring. The medication that you're talking about, what does it do for a patient and myself that's been on Plaquenil and Imunin and Cellcept for a very long time now and still flaring? How is this medication going to change my life?
So it's, of course, a great question, and I'm hesitating because I don't want to overstate what I have. I don't want to go beyond it, and we are at the very beginning of this. I think that the standard treatment that your doctor is probably, Plaquenil is a great drug that's very well tolerated, so I think the advantage of that is that we can leave patients on it for a long time, and it may help the patients, and there's very little downside to it. All of the other drugs that we use for flares in kidney disease, and I do want to distinguish that because kidney involvement is often more severe than other manifestations of lupus, but all of the drugs that we use for kidney disease are pretty potent immunosuppressive drugs, and when we add them to Plaquenil, I think we always add them with an endpoint in mind, and sometimes that endpoint is years away, or months away, or years away, but these are not things that you want added on indefinitely. And so the hope would be that this would be a new approach to add to Plaquenil, but would be closer to the Plaquenil paradigm where it wasn't so stressful to add it. You could leave it on and it would do good, but you're not exposing the patient to cancer risk and infectious risk, and from the very beginning hoping to get the drug away again.
Would I still have to take the prednisone? Would this eliminate me having to take prednisone as well?
So this is very far away from being an option for you, and I want to acknowledge that. And right now we do typically include prednisone in all of the treatments for lupus nephritis, and it's one of these things. Every time we start patients, even on Cytoxan, the biggest most powerful drug we have, we start them on prednisone, and we always ask the question, do they really need the prednisone? We're giving them this other great drug, and to some degree we're kind of historically stuck. They did the studies this way, and we're all afraid to vary from what they did in the studies. And so I know that prednisone of all the drugs is the one that people would like to get rid of, and I'm not sure I see the ability to get rid of prednisone anytime soon, unfortunately.
You stated that annexin A2 is being studied now specifically for lupus nephritis. Do you see the potential, or will you be increasing the studies into perhaps tubular renal acidosis or interstitial nephritis as well, or only glomerulonephritis?
The kidney tubules produce very, very abundant annexin, so it raises the possibility that it's equally or even more important in the tubular interstitium. And we have studied some more purely tubular models of disease. Actually, Dr. Silverman and I were discussing this briefly earlier. As nephrologists, we do make distinctions between glomerular diseases and tubular diseases, and I'm not sure we know 100% that they are distinctive, because first off, when we do a kidney biopsy, we are trying to get the glomeruli, so we may we may not get the proper impression of what's going on in the tubular interstitium. It is my belief that because the tubules make so much of this, and because the tubules can be such an important part of these different kidney diseases, that it may have a role there, but I don't know that yet. All right, well, thank you very much for your attention today.
I appreciate the invitation from Peggy and Diane to address you all today. I'm a rheumatologist. I spend most of my time in the laboratory. I have a terrible sense of humor, so I'll apologize in advance, but I spent more than 30 years of my life in San Diego, and I found that even though I have a passion for my patients and working towards earlier diagnosis and better diagnosis and understanding pathogenesis, San Diego is a little bit smaller than New York City, and so I ultimately, about 4 years ago, moved to New York, because the idea is if you want to study a human disease, you should go to a place that has more humans. And it's true. It is true. So let me just put this into perspective, because I've been thinking about lupus for a long time, and you know, I teach graduate students and medical students. And I have to say that lupus is one of the most challenging of all diseases, not just to conceptualize, but to try to explain it to my students. And I think I'm kind of getting it over time, but it's only taken, what, 25 years or something like that, 30 years. And, you know, particular interactions, experiences that I've had with patients have taught me a lot. And I have to say that one of the great things about being a rheumatologist, and I try to tell my students why they should be rheumatologists too, is that we talk to our patients. And I truly believe that every lupus patient is unique and special, and they'll tell you exactly what's going on with their disease and what you can do for them if you can find the time to talk with them. And I was just thinking about this because there was a day, and this was something like, oh, it's embarrassing, about 20 years ago. I was in a room and I was talking to a patient that I'd followed for a couple of years, and I don't know, we got off topic, and she mentioned that her sister was coming to visit. She hadn't seen her. Her sister had moved out of town, and we were chatting, and she said to me that it was her identical twin. And I said, really? And I have to say that from the perspective of a scientist who's interested in human disease, genetics is very, very important. And we know now that there are at least 30 different genetic intervals and variations that in different populations can predispose to the development of lupus. So that's very challenging. We try to actually see whether those genes may fall into certain patterns and that there are common pathways. But what an accident of nature. You have somebody who more or less is a clone. So the person I was talking to, unfortunately, had lupus, was very severe. It turned out that she actually deteriorated over the following year. We ended up starting a trial where we drew blood from her, and when her sister came to visit, we got blood from her as well. And I have this mental image of the two of them when I met her sister for the first time, and unfortunately, her sister was the lupus patient was very, very sick, and she actually had destroyed her kidney. She had developed glomerulonephritis, and she was actually on dialysis. And in my mind's eye, I was talking about this last night. I really don't talk about this very often. But I remember her sister visiting, and we were all in the hospital, and her sister visited when her sib was on dialysis. And it's such a dramatic scene, I have to say, because your life's blood is passing through this machine, which is really washing it because your kidneys have failed. And all of the damage done by the disease as well as the therapy, you could see on her face and just in how she held her body. And her sister was standing next to her, and she had her 1.5-year-old baby in her arms. And you might not know that they had anything to do with each other, but if you sort of put your eyes out of focus, they're the same person. One's sort of before or after, one's affected by lupus and the other isn't. And it was really amazing. And I have to say that not just the ravages of the disease and not just the challenges for earlier diagnosis, better therapy, but what the heck was going on? It's just amazing. And so identical twins, as we say, discordant for lupus, over time, your twin may well develop the disease, but at most, it's somewhere between 25% and 50% get the disease. So it can't all be genetics. Something else has to be going on. What is it? We spend literally millions and millions of dollars with the latest technologies trying to understand genes at a greater and greater depth. We've solved the human genome. We actually know the maps for the human genome. We are looking at all the different variations. We've done genome-wide scans with hundreds and hundreds of lupus patients. But there's something that we're not getting, something that we're missing. So I think that's actually what drew me to this topic, which I'll describe to you. And it's called the microbiome and lupus. And this is sort of an overview of the way that we think about things. Is that we all start out with a set of genes. We inherit them from our parents. They're sort of mixed up a little bit. Some may predispose to some diseases, and it'll never happen. And I have to say, in studies of genetics of lupus, we actually think that some genes increase our immune responsiveness that may well predispose to lupus. Maybe they were selected over the centuries because maybe our ancestors survived the plague or malaria or tuberculosis because our immune system was better and stronger. But now, in the world that we live in, maybe there aren't as many threats. There's what something is called the hygiene hypothesis, which is that actually the world is too clean. We don't really have to defend from all these microbes. So the most active immune systems start to attack itself. So this is on a timer or something. But so the genes, not every time, will turn into the disease. In some people, they'll develop into an autoimmune condition. We have autoantibodies that can actually bind to DNA in lupus. We think this is a major marker of disease and actually leads to the kind of kidney disease that Dr. Thurman was discussing. So what happens in between? Why does one twin get the disease and the other doesn't? We don't really know. It's a big question mark. We wonder whether or not it's something that we're exposed to. Something triggers a development within the immune system. And I have to say that I spent many, many hours, many weeks, I couldn't even tell you how much time, studying the immune system of these twins. I looked at the blueprint for their immune responses, and I looked at their antibodies at a molecular level, and you could see that they actually had the same roots. And one of them actually had developed into some very strange antibodies that clearly were DNA binding. We actually looked at them in great detail. But her sister's immune system got activated but didn't go any further. What is it that actually makes you go that extra bad mile? We think that it may be something just accidental, but it may be something in your environment. And maybe it's a virus that you're exposed to that you think you resolve, but it causes something smoldering. But to tell you the truth, the microbes that we're exposed to on a daily basis aren't just viruses. We actually carry them with us everywhere we go. And that's what I'm going to actually tell you about. So we all contain a zoo. We call them commensal bacteria because bacteria are not necessarily bad. In fact, they don't know if they're good or bad. They don't think about it much. We actually need to have them in us. And so the perspective is intriguing because we all think that we're so hyper-hygienic. We all go to CVS and buy the detergents and the deodorants and everything else to wipe out every aspect that we actually are basically animals, I think. But on us and in us, for every single human cell, we have 100 bacterial cells at least. So, I mean, some people would argue that they're the ones that are really in charge. We're just carrying them around and doing heavy lifting. And within our bowels, which actually has the greatest number of bacteria, there are 1,000 different species. So this is a very, very complex ecosystem. And the reality is we didn't even have the slightest understanding of what was going on within us because we didn't have the right scientific tools. And so all of us has the experience. We have a sore throat at some time. We go to the doctor. They do a little swab. They do a little culture. It grows. And maybe you have strep throat. The reality is at least 30% of the bacteria in you and on you cannot be grown. And it's actually a little bit artificial because they have media that they've developed for years or decades that are really good at growing one thing, but what are the other things that they can't grow? So in fact, now we have a technology so we can do these surveys, we can inventory, and we can get a better idea of what the relationships are between these different bugs and potentially how they contribute to health and disease. This is just sort of a mini segue. The point is, we always think about, we're being assaulted by people coughing on the subway or, somehow or another, don't touch your face or whatever. Forget about it. You're carrying within you so many microbes, it's just crazy. And, within the first day of life, you become colonized. This is the lumen of your bowel. And the point is that there is a battle just to maintain where your immune system is actually perched and ready and reacting and very activated, just trying to keep everything cool within you. And the question is, does that actually fight it to a standstill? Is everything really good? Or maybe it results in a very high level of activation that actually is the source of lupus. And in fact, in passing, I think what Peggy was actually telling us all about the interferon signature. Where the heck does that come from? We always talk about this in response to viruses. Bacteria can do it as well, and in fact, within different parts of the immune system within your abdomen, there is an interferon response, and one hypothesis is that it's really some bad bacteria that are having a bloom or a growth that are actually causing this interferon signature that may be the trigger of lupus itself. From another perspective, we know what the human genome is. We've sequenced the whole thing. I think it's 2 or 3 billion base pairs. Sounds like a lot. The reality is humans are just about 23,000 genes. That's it. It's not that complicated. The bugs that we carry in us have more than 1 million. So think about the complexity. And these are not just little pieces of blueprint that you have floating around inside you. They're being produced. They're doing things. They are having a metabolism. They're actually in relationships with one another. And the point is that our microbiota, which is what this community is called, is the major determinant of actually the food we eat, what we actually absorb, because the bacteria may actually get the better part of the deal. They actually determine, when you take medications, what parts are absorbed, what are metabolized, and what's actually active. So it is largely the case that some people may be responders and others are not, just because of the way the bugs in them are metabolizing the drug. And you can take an antibiotic and you can alter everything. And I have to say a tremendous example is Coumadin. Coumadin that is used to treat antiphospholipid syndrome and thrombi. It's related, something like 10% of lupus patients have it. It's actually an inhibitor of vitamin K metabolism. Vitamin K you don't make. The bugs in your gut do. So you shift that metabolism and you can end up maybe having the right balance in your clotting factors, or maybe actually being at risk of a terrible bleed as a complication. And every medication you take can be affected. There are papers that suggest that, in fact, the effects of the hormones that you make, estrogens and androgens, that it may be the bacteria that make the active forms. And as I was suggesting, what your immune system is doing and how activated may really be determined by the bugs within you. So our question really is it possible that the microbiome is contributing to the initiation of the disease, or maybe it explains why people flare? And so I just thought I'd show something complicated. We extract DNA from it, and we actually determine the blueprint. And we actually can inventory and see the distribution. And our first analyses suggest that lupus patients that have active disease seem to have fewer kinds of bacteria. And you go, how does that make sense? Their bowels are no shorter, they're no littler, they're not less active. But it seems as though some people, when they have flares, actually have a disproportion, a dysbiosis, an imbalance and a bloom of certain bacteria, which may well explain or at least contribute to flares or other changes in activity. We can actually use the kind of sequencing technology that was developed for the human genome to determine the microbiome genome. And we can use that both to look at the distribution of bacteria as well as their own metabolic genes that are involved. And through different studies, we can look correlatively, but we can also hope over time to identify what might be the protective bacteria versus those that are more likely to cause disease, and see if we can shift them. Antibiotics are a pretty primitive way of doing it. It may actually be that the products of the bacteria themselves, what their metabolic products are, the things that influence us. That may change things. So it's really a whole new field. It's really very fundamental. And I have to say that we're trying to coordinate studies both in humans as well as in mice so that we can actually test hypotheses. But I think there are tremendous opportunities for the future, and they may actually provide some practical approaches. So I just want to thank the Lupus Research Institute for supporting our studies. These are really, as we say, high risk, but I think that we're certain to learn a lot, and it's going to better explain how our immune systems work in health and in lupus. Thank you.
Hi, good afternoon. My question is, if you work harder to keep your gut cleaner, does this work with the information that you provided to us today?
So the question is, what does clean mean? And so it's really what the balance that's induced. And I have to say that there are examples on the kinds of diets can actually change the balance. I think that we don't have definitive answers, but there are, whether or not people are red meat eaters, and I have to say that actually has been shown to change the distribution of bacteria, and there's arguments that might be pro-inflammatory as opposed to people who eat simple sugars, which obviously some bacteria like better than others. And we're just learning about not just what enables an expansion of some strains versus others, but what the implications are for health. So there's a lot of, I'll give you actually a concrete example. So one of my mentors in this field is Martin Blaser, who's at NYU. And he actually is funded to investigate causes of obesity in the United States. And you'd say, well, what does this have to do with anything? So he points out that, in fact, the introduction of antibiotics routinely in the animals that we eat, actually, if you treat a chicken or a cow for a brief period of time, a few weeks, with antibiotic, and then you take it away, that animal actually ends up gaining weight and getting heavier and fatter for the rest of their lives, even though the exposure is brief. And he would argue that it's changed the balance of the bacteria, and he actually would also argue that the introduction of oral antibiotics that are given to kids routinely, starting in about the 50s or 60s, might actually explain perhaps the epidemic of obesity, but also the epidemic of autoimmune disease. More than 6% of Americans are now affected by one autoimmune disease or another. So there is some qualitative evidence. There is some evidence to suggest that certain bacteria are expanded. Another report out of NYU found that, in fact, patients that were within their first few weeks of arthritis that was diagnosed as rheumatoid arthritis had an expansion of a single strain of bacteria called Prevotella copri. And so is that really the trigger? Is that the fuse? Bad genetics? Just the wrong exposure? Time will tell, but we're having some initial findings that we're trying to translate to lupus research.
And as you know, there's a recent paper that came out about intestinal dysbiosis in lupus. And so about I can't pronounce these, but firmicutes and bacteria.
Firmicutes or something.
Yeah. And so, and they found that the ratio was much lower in lupus patients than in normal. And obesity, it's actually much higher.
Yeah.
So maybe you could just speak to that a little bit in the context of what you were just talking about your friend.
Well, a lot of these are observations, and I have to go from doing the inventory and figuring out what the balance or imbalance is to actually understanding the mechanisms causing it isn't really clear. And I have to say that there are some protective bacteria that are associated with ulcerative colitis on one side, but actually I think I'm trying to think what the other inflammatory disease was on the other, so I can't actually tell you. Things are relatively early. What is today? Today is the 21st. Before the American College of Rheumatology meeting, we're going to have a meeting that's all about the microbiome and inflammatory and autoimmune disease, and it actually was meant to bring together experts in the field and to focus interest in arthritis-related diseases. So maybe it'll all come into focus, but it's a relatively young field at present.
Great. Well, I'll see you at these.
Okay. Okay, almost got off.
Hi. I think I heard you say that with lupus patients, there seems to be less bacteria than...
It isn't, so these are very preliminary results. It actually has to do with the diversity or how many different types. So the space is the same, but it looks like there's an expansion of some bacteria so that there's less variation. It's as though if bacteria were meant to have an equal distribution in the alphabet, for some reason, lupus patients only have the last 6 letters. So they're more expanded, they're overrepresented, and maybe those are the guys in the black hats that are causing inflammation that's then affecting the rest of your body.
Well, just a wild thought. Is there any thought that maybe putting more bacteria types into a lupus patient could?
Yes, yeah, and so there's a paradigm. Pseudomembranous colitis is a disease that people get when they get too much antibiotics of the wrong kind and they get an imbalance and they have an expansion of Clostridia difficile. We're really good at these Latin names. It doesn't really matter, but it makes a toxin that really blows apart your bowel. And the point is that we have other antibiotics that can get rid of that bug, but it may never go away. So you treat and treat and treat with antibiotics. They're actually doing, so to speak, transplants that will change the balance of the bacteria. I have to say that it's becoming accepted, and actually I believe that it's under FDA evaluation, but there may be easier, more practical ways of changing the balance. But there is a precedent that that actually makes sense. And the whole idea of using one microbe against another is logical, in fact, because it's a jungle out there.
Well, I'm really excited to be here today. I've been working on this project. Our 1-year anniversary of the LinkedIn site was October 3rd. So it's a great month to be here and bring everybody up to date about what's been going on. So I'm a chemist by training and a pharmacologist, and then I meandered into immunology. So I thought it was appropriate to have the medicine bottles on the side of my slides. So I'm talking about this project called LRXL-STAT, which is all about finding compounds that are commonly used, either FDA approved or GRAS approved, generally recognized as safe, and looking at the evidence to see whether they could be used for lupus. So, but first I just wanted to start out and tell you a little bit about myself. I'm a basic translational scientist who's worked on lupus for my whole career. And Peggy talked a little bit about that in the beginning. Basic meaning I started out as a chemist looking at interactions of molecules. I went in and got a master's degree in pharmacology, so studying the details of drugs, how they work, all their mechanisms of action, and then went into immunology because I wanted to apply my knowledge to a whole system, came across lupus, and I haven't looked back. That's what I've been working on ever since. So I'm the scientific coordinator of this drug repurposing program, and I've I spent about 6 months collecting and evaluating treatment ideas through that LinkedIn site, which I'll tell you a little bit more about. And we reached out to patients, physicians, scientists, companies, and I got suggestions from all over the world. I think the farthest suggestion away I got was from Tasmania. I'd love to make a map sometime and show where all the suggestions came from. And I've spent about 1.5 years on this project overall. So just to give a little background and put in context, a number of you have lupus, a number of us work on lupus, but I think something that we all need to keep in mind, and as scientists sometimes we forget, is that it's more than just the molecular interactions, it's more than just the signal transduction mechanisms, it's a whole dysregulation of your body and your immune system affecting all the organs of your body. And you can have all aspects of the disease one at a time. Some patients cycle through them, but we need to look at the whole body, the whole system, when we're thinking about what's going wrong. And certainly in my position, when I'm trying to find things that could be repurposed in lupus to bring to the patients to bring relief, that there are many systems and not just one trial or one drug is going to be good for everybody. My goal is to find different interventions among all sorts of categories and to make sure that we have some trials going in all the different manifestations of the disease that I'm outlining here. So you know the characteristic skin, the butterfly rash, that's often how a lot of people realize that they have lupus. Pleural effusions, the lungs, your heart, your kidneys, your joints, Raynaud's phenomenon. And just to keep this in mind, that it's the whole person, it's the whole system. And as Greg has pointed out, it's also a balance of what's inside you and the bacteria. And that's an up and coming field, and one I think that Greg's going to make great progress on. I've known him for about 20 years, and once he digs into something, he doesn't let go until he solves it. So lupus affects women and minorities disproportionately compared with the general population. This is another point that we're trying to keep in mind when we evaluated all the treatments that were suggested, thinking about the trials. You know, sometimes as scientists, we lump all lupus patients into one group, and they're not. They're very different. The African American disease is much more aggressive. It occurs at a much younger age, and the genes that are involved in that are very different from the Asian disease or from the Northern European Caucasian disease that usually doesn't happen until you're in your late 30s or early 40s and is usually much less aggressive. Lupus nephritis in kidneys is a much more prevalent manifestation in the African American community, and the skin lesions are more prevalent in European Caucasians. So we need to keep this in mind, and again, I've been focusing on this and trying to find different treatments and trials that will fit all of these various manifestations so we can attack this, as I like to say, in a quantum way, in a head-on way, and really look at what could work. Now, I know, and I just did what I said most people do, which is go back and forth on the clicker. I tried to practice that beforehand. So despite for the desperate need for new treatments, as everybody knows, there's only been one drug approved for lupus in the last 50 years, belimumab. Most of the drugs people are taking have been used for a long time. They have a lot of side effects. One of my missions has been to try to find treatments for lupus that have a lot less serious side effects and that can be tolerated, and also to look into drug companies, into their pipelines, and try to find things that are being developed for other diseases and talk to the companies and bring them over to lupus patients. I had to leave the Lupus Industry Council meeting a little bit early to come talk to you guys today, and that's what we were just discussing. It's a great council. There are about, I think, 15 companies on it now, and they're all committed to sharing their resources, both personal and financial, as much as possible to work with organizations like the LRI and the ALR and the LFA and all of the other ones to try to make the trials as targeted for the different patients as they can, because that's a real problem. So how's the community tried to find new drugs? So I just wanted to touch on that for a couple of minutes. As Peggy said, organizations like this support basic research, and that's very important. They support translational research and clinical research. That's what I've done for my whole life. The pro is it's very productive, but it takes a long time. You know, actually a lot of the grants that the LRI funded in the beginning, it's actually taken a shorter amount of time to get to result, and that's because of their dedication to mission, their interactions with their investigators and cultivating that relationship. But still, even in that situation, a discovery that was made 10 years ago by Dr. Peggy Crow, who's the scientific director of the ALR, has really only come to fruition now, which is 10 years later, and that's really fast. So how can we find some things now within the next 5 years at least? And so just to even bring that home more, I found this slide about what is the typical development of new drugs in pharma or biotech or different kinds of process. Well, there are many, many stages going from very basic research to optimizing, to putting into animal models, and then doing the different phases until it's launched, just like the drug that Peggy was talking about was in phase 2B. And it costs a tremendous amount of money. It takes over 13 years on average, is an article that was published last year, and $1.78 billion with only a 9% success rate. So we tried to think out of the box and think of some way that we could bring some new treatments to patients in the near future, and that's what the Lupus Drug Repositioning Program is all about. And initially, we're focusing on repositioning drugs that were FDA approved or were generally recognized as safe, which is a lot of the nutraceutical industry, and to try to bring those into the initiative and to see whether they would work for lupus patients. So somebody else came up with the name, not me, but it's kind of like my first name, Amory. Once you get it, you'll remember it, LRXL stat. So what is LRXL? It means the lupus treatment list, RX being treatment. And it's a cooperative initiative. It was launched by the LRI and the ALR as a partnership. And the goal was to fast track new potential treatments for lupus. And if you have a pen, write down this web address or just put in LRXL stat into Google and the site will pop up. You can click on it, say connect, and I'll accept your connection. We'd love to have more patients on the site. Actually, patients are very underrepresented on the site. So I welcome all of you. Please tell your friends about it, and you'll get up-to-date information on a daily basis about what's going on with the initiative. So what is drug repurposing or drug repositioning? Other people call it drug reprofiling, retasking. It basically means taking a compound or treatment that was used for another disease and using it in lupus. So what is repositioning? It's determining whether a drug approved by the FDA for one disease is effective for another condition like lupus. And so I've had a lot of people ask me, you know, what are some examples of that? Well, a lot of you know examples that you don't even really realize. So Rogaine, that was developed for a completely different condition of which is being sold for now, which is hair loss. Requip for restless leg syndrome. Lyrica is now being used for fibromyalgia. And probably the most famous one, Viagra, which was being developed as a heart medication, and we know that men are buying it for another reason. So what is drug repositioning? Well, it's taking something like that's been FDA approved and using it, you know, for another condition. So actually, most of the drugs that lupus patients are taking technically fall into the repositioning or repurposing category because they've never been approved by the FDA for lupus. The only two drugs that have been approved by the FDA for lupus besides belimumab are glucocorticoids and hydroxychloroquine. All of these drugs, many of which I'm sure you're on, MMF, Cytoxan, azathioprine, methotrexate, and rituximab, they've never been approved for lupus. They were just used for that disease because of experience and publication. So it's not a new phenomena, and it's a phenomena that we've tried to hone and to bring to fruition to actually bring some more modern drugs to lupus patients. So I showed you this outline and how much money it costs on average to get to a new drug in pharma. And this is where drug repositioning comes in. And that's looking at drugs that may be already in clinical trial for another disease, or working with companies like we're doing in the Lupus Industry Council. They're coming to us and saying, we have something that we're developing for MS, but our scientists tell us that the signal transduction pathways, or how the cells talk to each other, is very similar in MS and lupus. Let's talk about it further and see what we can do. Come to somebody like me and my initiative with the cadre of people I have working with me to try to go through it detail by detail to actually see whether it would be applicable for lupus. And then, you know, as I've said over and over again, the goal is to make it available for lupus patients, and to do this as quickly as possible, we hope within the next 5 years. So just to summarize, the Lupus Drug Repositioning Initiative was initiated by the LRI and the ALR to address the slow pace of development of new therapies. And it's focused on fast-track identification of new treatments for lupus, and they're going to be tested in a program of small, biomarker-rich or science-rich clinical trials that will be done. And that part of the program is called STAT, we want it now, SLE, Treatment Acceleration Trials. And, you know, one important aspect of this, which Peggy touched on in the beginning, is we're going to have many trials going on in parallel, testing drugs of many different kinds. So you may have seen my poster that was hanging up when you came in today. We have different categories. We have biologics, which are like rituximab. We have small molecules, which would be like Cytoxan. We have cellular treatments, which may be like stem cell transplantation therapy. And we have alternative and complementary therapies. And there are many examples of those, nutraceuticals, as well as different personal interventions to help patients cope better with the disease at hand. And this program is going to be starting up in 2015, and we'll be announcing a number of trials within the next 4 to 6 weeks. So keep your eyes posted on the website. So the initiative, we've wanted to make it as open and transparent as possible, to have active outreach to all aspects of the community, patients, scientists, physicians, and I didn't even expect it in the beginning, but companies came to us through the site as well as with their ideas and with their compounds. And to gather ideas about what FDA-approved drugs could be repurposed, but people also suggested things that were in their pipeline and could be repositioned. I personally, with a little bit of help from some students, searched the entire database of drugs approved for human use in the United States. I had to get a new prescription for eyeglasses after I did that. And we also incorporated suggestions of a panel of experts convened by the LRI, ALR, and Lupus Therapeutics about 1.5 years ago. I also looked through the literature and looked for agents that have been used in murine models and have been effective, whether it was a gene that was altered by getting rid of it or giving the animal too much of it, or by treating the animal with a compound. So LRXL has really been a venture for me into social media. I did a little bit of Facebook before, and my friends told me I should do more to connect with people all over the world. So I decided to dive in and make this LinkedIn site for the project. And as I mentioned, our anniversary was just a couple of weeks ago. So if you put LRXL stat into Google, or you can put the website in directly, you can go and find it. We now have over 575 connections made up of patients, researchers, and people in pharma and biotech. And I'd love to grow that even more. I mentioned we especially need to have more patient voices in that community. And there are representatives from all the major lupus organizations that are part of it and are trying to contribute in a team manner to bring this initiative to fruition. So it's very interactive and transparent. Like I said, it's, you know, as a lot of you know, you can set it up on your phone, and when I put a little message, you'll get a message and see what's going on. We have over 625 accesses a week. It's been an incubator for novel ideas. People have had their own internal conversations about experiments that should be done, companies that we should talk to, and that's been very successful. It's generated over 100 treatment suggestions, including 16 from lupus patients, some of which have made our top priority tier list. So it's been very useful. And over 25 pharma and biotech companies came to us through the site and inquired about being involved in the project and potentially have their compound go into trials. So how did we actually do it? Well, first of all, anything that came through the site, we looked at in detail without any prejudgment. And Dr. Lipsky and I, from our own reading, we rolled out things that were FDA approved or things that were already in development and going into trial by a pharmaceutical company because we didn't want to duplicate efforts. So we looked at why should we do it, the rationale. Does it make sense? And we really focused on safety and availability. So if you looked at the poster, the scoring system goes from a -16 to an 11. Belimumab, rituximab, all get around a 5. So anything in that range automatically got into the high priority list. But we weighted safety very heavily. So you could get sunk a -5 from your bad side effects. You could get sunk by 3 or 4 points if it was dangerous for lupus patients to take because the kidney processed the drug. So we really took safety, availability, and how it was metabolized very seriously when we're ranking these treatments. We prioritized them after reviewing the evidence, and we identified high priority candidates. And we discussed the candidates with experts last April. And so this was a group of people from all over the world that had expertises in all the different fields. And I got up in front of them for 8 hours, presented all the drugs, and I answered their questions. I haven't been more exhausted in my life since when I got my PhD. But it was well worth it. They gave their thumbs up to high priority candidates. And some of those we're going to be putting into trials very soon. So this is a stick figure of how it worked. We got community suggestions through the site. We had an expert meeting in the beginning. Dr. Lipsky and I searched approved drugs and saw ones that were active in lupus mice. We came up with 157 things to look at, which, wow, that's amazing. I was thinking when the site started, if we had 50, I would be really excited. And we got 3 times that much. We went through them very systematically, and we developed a scoring system that I mentioned where we really watched out for side effects and for processing in lupus patients specifically. And then we got candidates to examine in clinical trials. And that's where we are now, is thinking about these trials. We have a number that we're going to announce at the American College of Rheumatology meeting in November. And with that, about 2 months ago, we initiated the Lupus Clinical Investigators Network. So these are doctors that take care of lupus patients all over the United States in every demographic. One of our missions was to make sure that patients had access to trials. Because I think one of the problems in the past is if you lived in New York City or you lived in Washington, D.C. or another metropolitan area, you had great access to trials, but if you lived, especially in the rural South, you didn't. And we want to try to fix that. The other thing that we've started is the Patient Partners Program, and you'll be hearing more about this. But this is within the trials, to have patients that will be resident to answer questions of other patients, who will be trained to explain the details of the trial to other patients and to be a great resource, somewhat in the role that Kathleen is playing for the FDA. And so we really wanted to grow and expand that idea. So the first part of this has been repositioning or repurposing. And the next phase, which I started last June, is looking at drugs during drug development that could be repositioned. And part of that I've done on a really global scale. So Dr. Silverman was talking about looking at the genome of bacteria, and he mentioned the genome of humans. So part of what we're doing now is gathering all the data sets that have ever been gathered from lupus patients genetically and their gene expression, and making a database and then querying it and asking specific questions for targets that could potentially be good for a drug. And so that's going to be our next phase. We started that in June, and those trials hopefully will be kicking off in 2016. So from LRXL to STAT, we want it fast, we want it now. And through the project, we've generated candidates for testing, and we really hope that we're going to bring new treatments to the patients that, like you and people all over the United States, regardless of their demographic or their market. I really appreciate your attention. I appreciate Diane inviting me. I'd be very happy to answer any questions.
So how are you working with rheumatologists to let them know about this, and how do we talk to our rheumatologist, you know, to find out which drugs we might try with them and be part of the clinical trial?
So there are a lot of questions in that, and so let me get to all of them. One is that the mother lode to go to is this website to find out all the information. Anybody who's interested in this initiative should just say connect, and I accept everyone as a connection. So that's how you get information. How are we reaching out to rheumatologists? We're doing it in a number of ways. Personally, just in the last 4 weeks, I've given 1 or 2 talks a week to different groups of rheumatologists. We also are doing it through the companies like the Lupus Industry Council, talking with the company representatives that are in the rheumatology offices to talk about the program. I'm also in the process of writing up an article that will summarize all of the information that will be submitted by the end of the year. And most importantly, I'm giving a presentation at the national meeting in November 14th through 19th in Boston. And that's where rheumatologists come to that from all over the world.
Hi. You mentioned alternative therapies. What alternative therapies have you found have helped patients with lupus? And are you going to be conducting clinical trials using alternative therapies or just drug therapies?
So we've taken alternative and complementary therapies just as seriously as the other categories. So biologics, small molecules, cellular and alternative and complementary. We hope to have 2 different complementary and alternative trials up and going in 2015. And for just confidentiality reasons, we're going to announce them at the ACR, so I can't disclose those now. One will be something that you take, hopefully, and one will be more of a behavioral or group sort of interaction. But we take them just as seriously as the pharma therapies. Well, thanks for everybody's attention. Hope to see you on the site.
Okay, so thanks everyone. I think what's so exciting about this project that, as was mentioned, was initiated as a joint project between the LRI and the ALR is the tremendous amount of collaboration and looking at things that are already on the market so that the safety has already been shown. So that these things would move to market much faster than some of the things that we heard about earlier, where you heard about, like the very preliminary work that Dr. Thurman and Dr. Silverman are doing. So this is a way to try and get some things to lupus patients faster because we already know the safety, which is one of the major hurdles in looking at new drugs.